Hey guys, welcome to the new video. Today I'm going to cover chapter 5, 6, and 7 of the exam 2 for human physiology. And so yeah, let's just go over it. Okay, for chapter 5, it covers the central nervous system and the peripheral nervous system. So, the nervous system and the endocrine system. These two can be compared. The nervous system transmits electrical signals or impulses to skeletal muscles and exocrine glands. It is wired, sending electrical signals through a distinct, highly organized pathways, and these pathways have interconnected parts. The endocrine system secretes hormones. These are chemical messengers into the circulating blood to distant sites in the body. And these glands are not connected. They are scattered throughout the body. So with the nervous system, this is the primary. And with the endocrine system, this is the secondary. Okay, so with the nervous system, it composes of the CNS and the PNS. The CNS, or the central nervous system, composes of the brain and spinal cord, whereas the peripheral nervous system is everything else but the brain and spinal cord. Other comparisons between the two systems. Okay, so I'm not going to read all of that. Instead, I'm going to read it in a summarized version. So in slide 3, in summary, the nervous system operates with a more direct and localized approach by targeting specific cells through neurotransmitter, while the endocrine system affects broader areas of the body, but only cells with the right receptors for specific hormones will respond. Okay, and slide 4 says, while the nervous system provides quick, short-term responses, the endocrine system ensures that the activities require long-term regulation or maintenance are controlled over extended periods. Both systems can influence the same processes, but to do so at different timescales. So in summary, the nervous system acts rapidly with short life signals, whereas the endocrine system takes longer to act but has a prolonged effect on the body. And then finally, slide 5. In summary, the nervous system and endocrine system are interconnected and are often influenced by the same body processes, but they do so with different mechanisms and timeframes. The study of their interaction is known as the neuroendocrinology. Okay. Now, slide 6. So I think with slide 6, I think it's better to explain in picture than actually reading in here. All right. Now, okay, so I already said this in my previous video, but I'll say it again. So imagine you are cold. Now your body telling you are cold is part of the afferent division. These are the receptors that goes into the brain and spinal cord from the sensory division. The central nervous system is part of the interneuron. Okay, now because you are cold, you decided to wear a sweater. So what happens is your brain and spinal cord sends signals through the peripheral nervous system, and then it goes to the motor. This is the effector. And then it goes to the somatic nervous system. This is the muscle block for the motor division. There's also a different part. This is called the autonomic nervous system. The autonomic nervous system is something that you can't control. You can control the somatic nervous system, but you can't control the autonomic nervous system because it controls it for you. It does it automatically. So, for example, you don't get to control the heartbeat. You don't get to control your arteries. You don't get to control your veins. You don't get to control your spleen. Your body automatically does it for you. And then with the autonomic nervous system, it splits into two more divisions. You have the sympathetic division and the parasympathetic division. So we know the sympathetic division as the fight or flight, and it is excitatory. Whereas parasympathetic division, we know it as the housekeeping repair. So it is inhibitory. But what Stark was saying in his video is that both the sympathetic division and parasympathetic division are both excitatory and inhibitory. And it just depends on which system you're talking about. And what do I mean by that? Well, let's put it this way. So in the sympathetic division, this is fight or flight, right? So imagine in a fight or flight situation where we are getting chased by a bear, our heart rate would go up and our stroke volume would also go up since, our heart rate went up. That's sympathetic. But the last thing your body would need is to filtrate urine from your kidneys. So filtration will go down, GFR will go down. So yeah, it's basically just emphasizing on you trying to survive. So your heart rate will go up, okay? Now in the parasympathetic division, imagine like you're sitting down like right now doing nothing. The last thing your body would need is to raise your heart rate, because why would you need to do that? You're just sitting down, right? So what would happen is heart rate will go down, stroke volume will go down, but filtration will go up and GFR will go up because you're not doing anything physical, right? So there is no need for the heart rate to go up. So in this case, this is basically saying that both sympathetic and parasympathetic divisions are both excitatory and inhibitory. It just depends on which system you're talking about and the current situation as to what's happening right now. Okay. So there are three classes of neurons. We have the afferent division, the efferent division, and the interneuron. So again, afferent is sensory. This is when it detects a stimulus from your receptor and it sends information to the central nervous system. This is the afferent, and it is usually at the back or the posterior slash dorsal of the spinal cord. The whole spinal cord right here is the interneuron. This is for processing. And then you moving your muscles or sending signals to do something with an effector organ is the efferent or the motor division, and it is usually in the front or at the anterior slash ventral part of the spinal cord. Glial cells do not send signals. They support interneurons physically, metabolically, and functionally. So you have four different types of glial cells. We have the astrocyte, oligodendrocyte, microglia, and the ependymal. So with the astrocyte, the functions include holding neurons together, guiding neurons during development, establishing a blood-brain barrier, repairing brain injuries, playing a role in neurotransmitter activity, and taking up excess potassium from the brain ECF. So right here is the astrocyte, and it does all those functions. Then we have the oligodendrocytes. The oligodendrocytes forms myelin sheath around the axons in the central nervous system. So as you can see here, it's forming myelin sheath around these axons. Then we have the microglia. The microglia are the immune defense for the central nervous system. They are scavengers or modified macrophages. So right here is the microglia, and they are the immune system of the central nervous system. And then we have the ependymal cells. The ependymal cells lines the internal cavities of the central nervous system. So these right here, these individual ependymal cells move the liquid in the central nervous system or in the brain. The liquid, by the way, that I was just referring to is also known as the CSF, or the cerebrospinal fluid. The CNS is protected in several ways. So we first have the cranium, or the skull, in which it encloses the brain. Right here, this is the bone, covers the brain. And for the spine, you have the vertebral column, and this encloses the spinal cord. And so yeah, now in your brain, it is surrounded by a stack of meninges. We first have the outer layer. This is known as the dura mater. This is separated from the dura mater by a narrow series of cavity termed the subdural space cavity. And then you have the middle arachnoid matter, which is right here. It is separated from the pia mater by subarachnoid space. And then you have the innermost pia mater. That's the last meninge. And the pia mater literally surrounds the brain. Your brain also is surrounded by the cerebrospinal fluid, so you can find the cerebrospinal fluid inside the subarachnoid cavity space right here. And the blood-brain barrier, which limits the access of bloodborne substances into the brain. So again, we have the skull right here, and then in the first outer layer, we have the dura mater. Okay, in the middle of the dura mater and arachnoid matter, you have the subdural space. The subdural space is basically just filled with blood vessels. And then you have the arachnoid matter and the pia mater. And then in the middle of the arachnoid and pia mater, you have the subarachnoid space. And inside that cavity is filled with cerebrospinal fluid. Okay, so the cerebrospinal fluid, or the CSF, is formed and it circulates. Okay, so the cerebrospinal fluid, what is that? Well, the CSF, or the cerebrospinal fluid, is just basically blood plasma with the big stuff filtered out. So in blood, you have the plasma, which contains 60% of the volume. You have the red blood cells, which contains 39% of the volume. White blood cells, which is 1%. Platelets, which is less than 1%. So the red blood cells, white blood cells, and platelets, these are form elements, and they get filtered to make CSF. So now we have plasma alone. Let's split that right here. Now plasma contains glucose, potassium, chloride, sodium, water, amino acids, hormones, albumin, and a bunch more. So any of these that get filtered out, they are made to go to the CSF. Okay, that's all you have to know. The big point saying is that cerebrospinal fluid, or CSF, are basically just blood plasma with the big stuff filtered out. Okay, so the CSF, it is produced by the choroid plexus inside the ventricles. It circulates through the ventricles, and from the fourth ventricle, it enters the subarachnoid space between the arachnoid matter and pia mater. The arachnoid villi is a space that drains the CSF back into the blood. So let's look at it in the picture. So we first have the choroid plexus of the ventricles, right here and right here. These turn blood into cerebrospinal fluid. The cerebrospinal fluid then circulates through the ventricles of the brain. Okay. Now, after leaving the fourth ventricle, which is around this area, the cerebrospinal fluid flows into the subarachnoid space. It does this because there's channels that leak out. And the channels that leak out, they lead to the subarachnoid cavity. And it's around this area, right? And as you can see, the cerebrospinal fluid goes right here, and then it goes right here. It basically goes around the subarachnoid space until it reaches the arachnoid villi, right here. The CSF eventually drains back into the blood via the arachnoid villi, right here. Cerebrospinal fluid. Again, they are found in and around the brain and spinal cord. It forms a liquid cushion that gives buoyancy to the CNS organs. It reduces the brain weight by 97% and prevents the brain from crushing under its own weight. It helps nourish the brain, and it may carry chemical signals like hormones, sleep, and appetite-inducing molecules. It is similar composition in blood plasma because it's just filtered out, and it is produced by the choroid plexus that hangs from the roof of each ventricles, and it is kept circulating by the ependymal cells of the ventricles. So the ependymal cells right here, again, they move the cerebrospinal fluid. So in summary, the CSF acts as a protective cushion, reducing weight and impact on the brain, while also playing a role in nourishing the brain and facilitating chemical communication. It is similar to blood plasma in composition, and it is produced continuously by the choroid plexus, and it is kept in circulation by the ependymal cells. So the formation of the CSF, we know that the CSF is basically just blood plasma that is filtered out. So as you can see, filtrates sodium, calcium, magnesium, etc., glucose, oxygen, vitamins, and ions. The flow of CSF in the central nervous system. So let's look at it step by step. We first have the capillary bed. This is in the choroid plexus. It filters blood plasma, and it flows inside. And once the CSF is made, it flows inside the lateral ventricle, down to the third chamber right here, down to the fourth chamber, and all that, and then down the spinal cord. The CSF is then pushed back up around the brain right here. Oop. And they get drained at the arachnoid villus, right there. At the arachnoid villus, CSF gets turned back into blood plasma. So we have the blood-brain barrier, and it is highly selective. The BBB, or the blood-brain barrier, is a series of capillaries that regulate the exchange between the blood and the brain. These capillaries allow a limited number of substances to pass from the blood to the brain cells. The brain needs a constant input of oxygen and glucose from the blood. So we have the blood-brain barrier, or the BBB. It is a protective mechanism that help maintain a stable environment for the brain. However, it is ineffective against fats, fatty acids, other fat-soluble molecules, oxygen, and carbon dioxide. So in this case, anything fat-soluble can go in, but if it's not, then it'll just bounce out like that. It is, in its structure, it is not completely in uniform. So this is referring to it not being symmetrical all around. In one side, it is thick, whereas in one side, it is thin. The blood-borne substance within the brain capillaries are separated by the extracellular space and neurons by the capillary wall, which is right here. It is a continuous endothelium surrounded by the relatively thick basal lamina surrounding the external face of capillaries. So right here's the basal lamina. And the bulbous feet of the astrocyte that cling to the capillaries, right here. These are the astrocyte, or the bulbous feet. So in other words, for a substance to go through, it needs to get to 3 layers to get to the brain. You have the continuous endothelium, the basal lamina, and the astrocyte bulbous feet. The function of the spinal cord and connections to the peripheral nervous system. So the CNS consists of the brain and spinal cord. We know that, right? And then the peripheral nervous system consists of everything else but the brain and spinal cord. But in the peripheral nervous system, it contains receptors, sensory neurons, and motor neurons. Sensory neurons are afferent to the CNS, meaning it sends information to the brain, whereas the motor neurons is efferent, meaning it sends signals away from the central nervous system. The spinal cord extends through the vertebral canal, and there are 31 pairs of spinal nerves. The cord ranges vertically from a large hole at the base of the skull, which is the foramen magnum. It's referring to somewhere right here. And the attached spinal nerves pairs are the 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. So what is this saying? Well, it's saying that in the spinal nerve, or the spinal column, you're going to have a pair of nerve in each of the little cavity space. So you're going to have a nerve right here, you're going to have a nerve right here, and they are counted in pairs. So there's 1 pair, 2 pair, 3 pairs, 4 pairs, and you're going to have 31 pairs in total. The spinal cord consists of the inner gray matter and the outer white matter. So again, the white matter is myelinated, whereas the gray matter is unmyelinated, and it is mainly for processing. Whereas for white matter, it's for speed, right? So let's look at my notes here. What we have is an ascending track. So this is information going into the brain, and then the descending track, which is information going out from the brain or away from the brain. We also have the central cavity right here within the middle of the brainstem. Inside the central cavity, you can find the cerebrospinal fluid, or the CSF. Again, these CSF thingies, they surround the brain, right? And they are located in the subarachnoid cavity. So yeah, that's one good thing to know. Then in the posterior side of the the spinal cord, you have the afferent division, and then in the anterior side, you have the efferent division. So the white matter of the spinal cord consists of tracts for vertical relay. All right, so we have these tracts. They can be ascending, which is the lateral spinal thalamic, and descending, which is the vertical cortical spinal. So this image right here, let's look at the ascending tract. Again, ascending, they are the spinal, thalamic tract right here. And as you can see, most of the blue, well, not most of them, because most of them are here, but just saying the lateral side, you can see a lot of blue, and these right here are ascending. They go to the brain. Okay, the ascending tracts range through the entire CNS, sending signals from the cord up through regions of the brain. And then we have descending. We have the descending tract. You can find them in the vertical cortical spinal. So right here, cortical spinal tract, yeah, around these range, yeah. Okay. It says here, descending tracts range through the CNS, sending signals away from higher brain centers down to the cord. All right, let's move on. So we have the gray matter of the spinal cord, which consists of 3 pair of horns. These horns are the ventral, lateral, and dorsal. And in the spinal cord, it contains the internuntial neurons too. Okay, what does that mean? Well, I got you. A nerve is a bundle of peripheral axon, and the spinal nerves contain afferent and efferent fibers. So one function of the association neuron is the spinal cord gray matter, and that is to integrate basic reflexes. They are a part of a reflex arc, which is a neural pathway that produces reflexes. So reflexes are involuntarily. You don't consciously do this. Okay. So let's put it this way. So imagine you are cooking something, right? I don't know. You could be cooking anything. And then let's say you accidentally touch the hot pot. Now, your finger has a bunch of receptors in there, and it detects pain receptors. And that pain receptor is activated because the stimulus, which is you touching the hot pot, sends a signal through your sensory tract division or your afferent division. Because again, afferent is information going to your brain, right? So information goes through here, this and that, and then boom, now we get into the interneuron. The interneuron does this automatically. It sends signals up to the brain, but it also sends signals straight up into the muscle without even reaching your brain. What happens is in this part right here where it just automatically does it for you, what it does is it releases an EPSP right here and an IPSP at the same time. So the EPSP tells one muscle to contract while the other is told to not contract. And so therefore, you automatically move your hands before you even know it. So that's what a reflex is. You just do it automatically. All right. And here are the 5 components of a reflex arc. We first have the receptor. The receptor is right here. And say a stimulus happens, right? We then have the afferent pathways. The afferent pathways, again, they go into the integration center or the CNS, central nervous system. Then we have the integrating center. Again, this is the CNS processing, what's just happening. And then it goes right here to the motor neuron or the efferent pathway. The efferent pathway goes into the effector, and the effector is usually in an organ or a gland. So again, the receptor detects the stimulus, the effector makes the response of the reflex, and the integrating center is within the gray matter of the spinal cord, and its neurons connect a given input from the afferent pathway to the proper output from the efferent pathway. And so yeah, basically now the other slides here that I'm about to show you are all just reflexes. And that should be it. And so then we have the peripheral nervous system. Chapter 6 contains the afferent division and the special senses. So again, the afferent division is information going to the peripheral nervous system and into the brain or the central nervous system. So the PNS or the peripheral nervous system has nerve fibers that can carry information between the CNS and body regions. Its afferent division sends information about the external and internal environment to the central nervous system. Okay, now these 3 dots are here. I'm gonna say it in here. So we first have the visceral afferent pathway. These pathway carry subconscious information from the internal organs, examples like your stomach or heart, into the central nervous system. You are not consciously aware of this information, but it's important for regulating functions like heart rate. We then have the sensory information that is conveyed to the level of conscious awareness. So sensory information that reaches conscious awareness can be classified into 2 types. You have the somatic sensation, right here, somatic sensation. Somatic sensation are sensory inputs from the skin, like touch, pressure, pain, or from proprioception, which is the awareness of body positions from muscle and joints. So proprioception is basically like you closing your eyes and you still knowing where your hands and feet are, or you know where your hair is. That's just you being aware of your body position. And we have the special senses, which is the vision, hearing, taste, and smell, which are complex sensory system. Perception is the conscious awareness of the external world. It is created by the brain from a pattern of nerve impulses that is sent to the brain from sensory receptors. The brain interprets an input, and human perceptions do not replicate reality. So what does that mean? Well, the brain's way of interpreting sensory information is not always an exact reflection of reality. So examples like these include optical illusions, magic tricks, or basically anything that deceives your eyes. Okay. Receptors are structures at the peripheral endings of afferent neurons. The receptors detect stimuli and carry information towards the CNS or the central nervous system. So in other words, you need to have a receptor to detect stimulus. No receptor, no detection, and no detection, no stimulus. So we have the classification by stimulus. Each type of receptor has an adequate stimulus, and the type of receptors are photoreceptors, which are the ones that respond to wavelength of light, and we have mechanoreceptor, which is sensitive to mechanical energy. Examples include vibration or pressure. We then also have thermoreceptor, which is basically sensitive to heat and cold. We also have osmoreceptor, which detects changes in the concentration of solutes in bodily fluids. Chemoreceptors, in which it is sensitive to specific chemicals, such as concentration of oxygen in the blood. And nociceptor, which is a pain receptor that is sensitive to tissue damage. So we have the classification of receptors. They can be classified by the location or the structural complexity. So in location, or actually by location, we first have exteroreceptor, which is outside the body. So examples include, like, I don't know, maybe like a ball hitting you, or the temperature is too hot, temperature is too cold. Examples like that. Basically anything outside the body. Okay. Now the interoreceptor is inside the body. So this is basically you saying my back hurts, or my foot hurts, or my head hurts. The receptor is located inside the body, responds to internal conditions. That's basically what it's saying for interoreceptors. And then we have proprioreceptor. So proprioreceptor are basically receptors that provides information about the position of the body. So examples include you saying, I know that I'm standing right now. I know that I'm sitting down. I know where my hands are. I know where my feet are. Examples like that. Just know the position of the body. Just knowing the position of the body. And then if it is classified by structural complexity, we have simple and complex. So again, the names is pretty self-explanatory. Complex, these are the special senses, associations, and the simple, which are modified dendritic endings of sensory neurons. Alright, so a stimulus alters the membrane permeability of the cells of a receptor. This leads to the production of graded receptor potential. So the receptor can be specialized ending of an afferent neuron or a cell closely associated with the peripheral ending of a neuron. This change in membrane permeability can lead to the influx of sodium ions, which produces receptor generator potential. The magnitude of the receptor potential represents the intensity of the stimulus, and a receptor potential of a sufficient magnitude can produce an action potential. This action potential is propagated along the afferent fiber to the CNS. So what it's saying is when a receptor is stimulated, it creates a receptor potential by altering the membrane's permeability. If the stimulus is strong enough, this receptor potential generates an action potential, which is sent to the CNS or central nervous system for further processing. Okay. So, by adaptation, receptors can adjust to sustained stimulation. With sustained stimulus length, the extent of a receptor depolarization decreases. This adaptation can be slow or rapid. So, what is it saying? Well, when a stimulus is applied continuously, the receptor's response decreases, meaning that the receptor becomes less sensitive to stimulus as time goes on. And this reduces the rate of action potentials being sent to the central nervous system. So notice that if you ever go to a room where there's like a specific strong smell that you can't seem to ignore, and then eventually as you keep going there and going there and going there, you get used to the smell. Like you don't even notice that it's there anymore. That's your way, or that's your body's way of blocking the receptor, in that it helps you, or it helps prevent you from being overstimulated. So we first also have the tonic receptor. Tonic receptors, they adapt slowly or they do not adapt at all, whereas phasic receptors adapt rapidly. So the Pacinian corpuscle detects pressure and vibration in the skin, and this adapts rapidly. So right here, let's look at the image right here. We have the Pacinian corpuscle. They detect vibration, and this can be found in the dermis of the skin. Okay. So afferent pathways reaching the spinal cord can be a part of a reflex arc, or can be relayed to the brain by sending pathways. So again, this is basically the analogy of you touching a hot pot, and the hot pot is a stimulant. So you touching it generates a stimulus. It gets picked up by the receptor right here, and then the receptor it goes to the sensory neuron. The sensory neuron goes into the interneuron, and the interneuron goes to the motor neuron right here, and it goes to the effector organ. But if we go back to the interneuron right here, what it's saying is that the reflex arc sends an EPSP and IPSP at the same time. But with the EPSP, it tells one muscle to contract, and the IPSP sends one muscle to not contract. Instead, they relax. So on the process of doing that, it sends signals in both the spinal cord and into the muscle itself. So before the information reaches your brain, you already move your hand, because it's much quicker to go to the to the motor neuron than actually going up to the spinal cord first and then sending information back, right? So yeah, reflex arc, they're significantly faster compared to voluntary reflex. Hopefully that made sense. So somatosensory pathways convey conscious somatic sensation. So a receptor detects stimulus, and a specific receptor detects a specific stimulus for each kind of sense modality. So for example, a skin detects temperature change and not the wavelength of light. That's the eyes. And so a first order sensory neuron sends signal from the receptor to the spinal cord. So this right here is the first order. The first order neuron synapses with the second order in the spinal cord or the medulla. And then the second order neuron synapses with the third order neuron in the thalamus. Each afferent and ascending pathways excites a defined area of the cerebral cortex. So, acuity for a sensation refers to the discriminative ability. The word acuity refers to the ability to distinguish different stimuli and detect fine details. So, put it this way. We have a point right here and right here. Although there's 2 points, these two afferent division of the skin surface have different receptor endings. So, for example, this right here only has one receptive field that is stimulated by 2 points of stimulation. The same distance as apart is felt in 1 point. However, this point right here has 2 receptor fields that are stimulated by the 2 points of stimulation. There's 2 points felt because the receptive field on the skin surface is much more smaller compared to this one continuous one. It's basically saying you have more receptive field. Okay. So this image right here shows a skin surface and a very sharp pencil. Now, the very end of the pencil that's actually poking you, you're gonna actually feel it more in the middle where it's actually being poked at the most, like it's stimulated the most. But in the outer side right here where it's not really being touched in a way, it is not stimulated compared to the one in the middle where it's actually touching the pointy sharp pencil. And it shows in this graph too. There's high amounts of sodium in the middle and then it spreads apart like that in the baseline levels of activities. So this image right here, I think it's better to explain right here. But before we go over that, I'm gonna explain slide 12 real quick. So the stimulation of nociceptors produces the perception of pain. However, there is also motivational and emotional responses that also affect the perception of pain. But in other words, there are still 3 types of categories of pain receptors. We have the mechanical receptor, which responds to mechanical damage. So basically you being cut. Or being hit by a ball in the face or something. I don't know. And then we have the thermoreceptor, which responds to temperature extremes. So remember that one story? Not even a story. I don't know why I said that, but just imagine you touching something really hot. That's temperature extreme or the thermoreceptor being picked up. And then we have the polymodal nociceptor responding to damaging stimuli. So this is literally anything. It can be a cut, it can be thermal, it can be chemical, in such that, I don't know, you're exposed to certain acids which causes irritation or something like that. It's polymodal, meaning polymodal are the body's general pain detectors, and they are ready to respond to a variety of danger signals from your environment. So again, with the EPSP and IPSP, we have the EPSP right here. So this is referring to one stimulus right here, activating, and then it activates another, and if it activates another, and activates another, and activates another. Now, if this happens, if EPSP happens, then you're never gonna know where that stimulus actually happened because all of these neurons are being activated all at once. You're not gonna know which one is actually the one that's being affected, right? So this is lateral inhibition. This is when the IPSP inhibits one side of the neuron to give you a more accurate perception. So let's say there's a stimulus right here, right? Now the lateral inhibition causes one neuron to not be activated, and therefore these other neurons that are typically activated are not activated no more. So it's like a domino effect. You stop one from falling, then none of them are gonna fall. So in other words, the EPSP right here illustrates how excitatory signals spread throughout the neuronal network, promoting action. And then right here, the IPSP or the lateral inhibition illustrates how inhibitory signals over here are sent to the neighboring neurons to refine and sharpen the response, preventing the spread of unwanted activity. Okay. So there are fast and slow afferent pain fibers. We first have the A delta fibers, which fires at the rate of 30 meters per second. This is the quick. A delta are quick. And then we have the C fibers, which fires at 12 meters per second. And this is relatively slow. There's a higher level processing of pain input. The ascending pathways for pain are in the somatosensory cortex, thalamus, and reticular formation. The brain has a built-in analgesic system. So in other words, pain is transmitted via 2 types of fibers, the type A delta fiber, which is responsible for sharp, immediate pain, and the slow C fiber, which are which transmit dull, persistent pain. The brain processes pain at different levels, and it also has a natural analgesic system that helps modulate and reduce pain perception. Okay, and that should be it. All right, finally, we're at chapter 7. So, chapter 7 is the efferent division. So, the last chapter, we went over the afferent division on how we learned how information is sent through the peripheral nervous system to the central nervous system. And now we're going to talk about the efferent division, in which the central nervous system and the peripheral nervous system sends information efferently, somatically, and autonomically. And also with the automatically, sympathetically, and parasympathetically. And so, yeah, let's just go over it. So, the autonomic nervous system innervates cardiac and smooth muscles. Innervate meaning control. They control most cardiac and smooth muscle, most exocrine glands, and endocrine glands. The autonomic nervous system only releases two different neurotransmitters on the structures it controls. So, the autonomic nervous system releases acetylcholine, which slows things down, and norepinephrine, which speed things up. And the autonomic nerve pathways has two neurons, a preganglionic fiber and a postganglionic fiber. So I already drew this, right? With the motor or the efferent division right here, you only have to know that somatic is part of the neuromuscular junction. So you moving your muscle is part of your somatic nervous system. And with the autonomic nervous system, that includes like your heart, your spleen. You don't control any of those. Your body automatically does it for you. Okay. So the two subdivisions of autonomic nervous system are the sympathetic and parasympathetic nervous system. So the sympathetic nerve fibers originate from the thoracic and lumbar. So it's your chest and your back. And its preganglionic fibers are short, and its postganglionic fibers are long, terminating at effectors. Whereas the parasympathetic nerve fibers originate from the cranial and sacral levels of the central nervous system. So that's your brains and your hips, or actually your head. Yeah. Its preganglionic fibers are long, and its postganglionic fibers are short, terminating at effectors. There are terminal ganglia near effector organs. Alright, so the autonomic ganglion is between the preganglionic and postganglionic fibers of an autonomic pathway. The preganglionic fibers of both branches of the autonomic nervous system releases acetylcholine. So, in other words, what this is saying is that in both the preganglionic fibers of both the parasympathetic and sympathetic division, both releases acetylcholine at their preganglionic fiber. Then, the parasympathetic postganglionic releases acetylcholine because they are cholinergic fibers. Releasing acetylcholine causes maintenance on the body. So in the parasympathetic system, releasing acetylcholine, they come in as acetylcholine and they end up releasing acetylcholine. Okay, whereas the sympathetic postganglionic fibers releases noradrenaline or norepinephrine because they are adrenergic fibers and they are made for fight or flight and they speed things up. So in the sympathetic system, in the preganglionic fiber, again, they both come out as acetylcholine, but in the postganglionic fiber, they come out as noradrenaline. To those who know, I'm just kidding. And norepinephrine. Okay, that's all we have to know. So with the parasympathetic division, again, that is part of your brain. No. Your sympathetic, is your thoracic and your lumbar, and they have short preganglionic and a long postganglionic. Whereas the parasympathetic is your brain and your sacrum, and they have a long preganglionic and a short postganglionic. The autonomic nervous system controls the involuntary responses of visceral organs. These responses range from circulation and digestion to sweating and urination. The visceral organs receive dual innervation. They are signaled by both subdivisions of the autonomic system. So the parasympathetic and sympathetic division control both. That's what it means to be dual innervation. Usually both subdivisions are active in controlling the activity of an internal organ, and this ongoing activity is called the sympathetic or parasympathetic tone activity. So with this point right here, it is adjusted depending on the body needs, like exercising. So with the sympathetic tone, if you're exercising, your heart rate increases, your blood flow increases, but in the parasympathetic, it dominates during rest and digest. So that's when you're resting. One system is dominant over another over a given time, but they can compete for control of an organ. The sympathetic dominates during the fight or flight responses, and examples include, the pupils dilate, sweating is promoted, heart rate increases, bronchioles dilate, so that's you basically breathing more oxygen. Motility for digestion is inhibited, so that's you focusing on just you surviving, so you don't really need to digest if you're trying to survive. Liver conversion of glycogen into glucose, urination is inhibited, and all that. And these responses prepare the body for emergency or stressful situations. So that's sympathetic, and they dominate during the fight or flight response. Whereas the parasympathetic, they dominate during quiet and relaxed situation. So the responses is signaled by the subdivision, and it is usually the opposite, in which it decreases the heart rate, or none. So no activity glucose production from the liver. The dual reciprocal innervation allows the precise control over the activity over a visceral organ. So again, this is the balance between two systems that allows the body to smoothly transition between rest and active states as needed. There are several exceptions, though, to this general rule of dual reciprocation innervation. Now I'm going to read it from this slide right here because it's easier. So while most organs are controlled by both sympathetic and the parasympathetic system, there are important exceptions, such as the blood vessels, sweat glands, and adrenal medulla, where only the sympathetic system is involved. And these exceptions highlight the specialized and focused control of the sympathetic system and can exert certain functions, especially during the fight or flight response. So again, this is the exception to the dual reciprocal innervation. Each autonomic neurotransmitter binds to several different receptors. So the cholinergic receptor binds to acetylcholine, whereas the adrenergic receptor binds to norepinephrine and epinephrine from the sympathetic postganglionic fibers. We know that. The nicotinic receptors are found on the postganglionic cells of all autonomic ganglia, and these ganglia are found where the preganglionic neuron signals the postganglionic neuron. The muscarinic receptors binds to acetylcholine from parasympathetic postganglionic nerve fibers. And then with this slide here, it's saying the activation of alpha-1 receptor usually produces an excitatory response in the effector, and the activation of alpha-2 receptors usually produces an inhibitory response. The beta-1 receptors are found mainly in the heart, and they can cause an excitatory response. The response to beta-2 receptors activation is general inhibition or inhibitory. Certain drugs selectively enhance or mimic autonomic responses. They are agonists, so they speed things up. While other drugs block autonomic responses, they are antagonists, so they slow things down. Autonomic activities are controlled by the different areas of the central nervous system. They include the spinal cord, medulla, hypothalamus, prefrontal, association cortex. So the somatic nervous system signals skeletal muscles. One axon and its nerve from the spinal cord terminate on skeletal muscles. So again, this is what we went over already. In the couple slides that I've been going over, I was talking about the autonomic nervous system, talking about the sympathetic and the parasympathetic. And now we're at the somatic nervous system. So the somatic nervous system is the neuromuscular junction. So that's your muscles. You controlling your muscles is the somatic nervous system. Acetylcholine is the terminal neurotransmitter at the neuromuscular junction, right here. This junction is where the axon of a somatic efferent neuron signals a skeletal muscle fiber cell. The axon terminates at a terminal button. The motor end plate is the specialized part of the muscle cell membrane facing the terminal button, and the somatic motor neurons are the final common pathway. Skeletal muscles activity can be affected only by acting on these motor neurons. It says right here, acetylcholine is released at the neuromuscular junction. So, basically, this is what I already went over, but I'll go over it again. Let's see. Yeah, this is basically what I just went over. So again, what we have here is an action potential. They open calcium voltage-gated channels. Calcium goes in, and the calcium binds to the neurotransmitter right here. The neurotransmitter that we know it as is acetylcholine. Acetylcholine exits by exocytosis into the junction right here. And then they bind into the ligand channels over here, letting sodium in, causing a small graded potential, which is called the EPSP. And the EPSP goes around the neuron, opening sodium voltage-gated channels, and that's when sodium goes in, depolarizing the cell, causing an actual action potential. And they spread over the muscle. So that's the neuromuscular junction. So this is basically what I just went over. An action potential at the axon terminal opens voltage-gated calcium channels in the terminal button. Calcium ions diffuse. By the calcium influx, acetylcholine is released by the exocytosis from vesicles in the terminal button. The acetylcholine diffuses through the space between the nerve cell and the muscle cell. The neurotransmitter binds to the receptor sites on the motor end of the plate of the muscle cell membrane. The binding opens channels for sodium influx into the ICF of the muscle cell. This produces a local current flow that opens adjacent sodium channels in the motor end plate. So the EPSP to the voltage-gated channel, which causes action potential. And then the action potential is initiated through the muscle fiber. So basically what I just went right here. This is basically the same thing except, an image. And you would need acetylcholine esterase to break down the acetylcholine that's being released here. Acetylcholine esterase destroys acetylcholine. We know that this terminates the action potential and responses of the muscle cell. The neuromuscular junction is vulnerable to several chemical agents and diseases. And examples include the two toxins, which are the black widow, spider venom, and the botulinum toxin, which alters the release of the acetylcholine. And the courier blocks the effect of release acetylcholine. And that should be it.